Okay, then let's get started. Welcome back. I have to remind you that the lecture is being live streamed and recorded and distributed. I hope we'll get those things edited and up in a form that's more usable, but at least we'll have the recordings available for people if they need to refer to things. So today I'd like to continue, start with a little bit of a review. There were some questions about some of the homework, and I want to go over some of the ideas and homework problems together, and make sure everybody is up on the same page with that. And I'd like also to have a chance for people to update us on their project ideas. And then I want to dive into how to actually do a little bit more detailed computer simulations with CompuCell. I don't know how much of the cell sorting we're going to get to today. I hope we'll see, depending on how things go. So if we go back to the last time, we started out using CompuCell software. Remind you, CompuCell consists of two pieces. An editor called Twinit, which has the specific feature in it that it has a lot of the simulation code that you might want to use to write simulations already templated in. And so that is very helpful, so you don't have to remember as many things. I'd say the biggest problem with Twinit is that there are a lot of menus, and so it's worth pulling the menus down and going through them step by step and seeing what's in them, because finding things is sometimes harder than using them. There is in the materials that was distributed with the course a walkthrough of all the menu hierarchy. And so if you print that out, you can look through that, maybe find things that are lurking there that you didn't know about. And then CC3D Player is the tool we use to actually run the simulations. And that gives us some control over what is going on in the simulations and more control on how they're displayed and how outputs are done, simulation outputs are saved. The actual CC3D simulations themselves consist of 3 or more sets of code wrapped together in a bundle. The main components are this CC3D markup language script, which we've been working with so far, a Python script called a Steppables script, which we'll work with today a bit, and then something called the main Python script, which actually doesn't, is typically not edited much by the user, which tells CompuCell how to orchestrate all of the various things it's doing. And the main Python script is mostly built automatically. We'll try to avoid having to edit it by hand. And there also are some additional files which have to do, for example, with the player configuration, which sometimes can be a little problematic. And also something that orchestrates resources, that .cc3d file that you'll see as well. Again, typically those are generated automatically by Twinit. You don't hand edit, although you can. So the first thing that I want to come back to, which we talked about, is the fact that CompuCell lives in a lattice. And so the very first thing you have to do in any simulation is to say how big the space in which you're going to do the simulation is. That's different from a off-lattice model, although typically models that use diffusion fields have to have some kind of specification of space, spatial volume they're running it. While it might be nice to have a spherical universe for CompuCell, the reality of simulation code is that simulations live in rectangular or brick-shaped domains. CompuCell does give you quite a bit of flexibility about lattice type. In particular, you could have either a square or hexagonal lattice. The square lattice is easier to do arithmetic on and to think about. The hexagonal lattice has some nicer features in terms of how it behaves. And another issue that you'll face, the square lattice in particular, is that Square lattices have what is called a Manhattan metric, which is distances in the left, right, in the north, south, east, west direction are different from distances in the northeast, southeast, northwest, southwest directions, in the sense that your distance to your nearest neighbor is 1, your distance to your next nearest neighbor is square root of 2. And that non-uniformity in distance, anisotropy in distance can cause some issues. And so sometimes you'll define the cells as being next to not just the nearest neighbors, but second neighbors, third neighbors, or even higher orders. And in CompuCell, there's a block of XML, which tells you that it's CompuCell. And the lattice specification, how big the lattice is, whether it's periodic in any given direction, and what the intrinsic range of interaction of the lattice is. And that's something we're going to run into again and again. CompuCell is intrinsically 3-dimensional. I want to make it 2-dimensional. I set Z to 1. The next thing that came up and talking with people in the homework, it's important to emphasize, I think it's important to reemphasize this, is that a cell, everything in CompuCell is, and this is true of any agent-based simulation, nothing exists unless you assert it. We don't get things for free. And in particular, when we talk about a cell, from a computational point of view, a cell is a set of voxels in our lattice, in our cell lattice. Each one of those voxels has an integer sitting on it. And all of the voxels belonging to cell number 66 have the number 66 sitting on them. And so, for example, here in this diagram, there's these blue-gray voxels, which are trying to indicate they all belong to the same cell. If that was cell number 66, the number 66 would be sitting inside of each of those locations. The next cell over, if it were cell 99, would have the number 99 sitting there. So a cell is, by definition, an index and the collection of all voxels containing that index. And that means that if I had a pixel over here in the corner that was also number 66, it would be part of this cell number 66. By itself, there is no requirement that cells be simply connected. The cell could consist of dust of individual voxels scattered throughout space. Making the cell connected is something that occurs as an emergent property of what you specify for the cell energy functionals. It's not automatic. Cells also have a second index, which is called a type. You can do much more complicated sets of cell definitions. But to begin with, at each voxel site, if cell number 66 is cell type mesenchyme, then there would be an associated flag that says that voxel belongs to cell 66 and has type mesenchyme. Another thing to remember is that while the voxels have individual identity, the boundary between two cells doesn't. And so the edges of a cell are not, while you can, CompuCell keeps track of them, they're not objects in a computational sense, in the sense that the cell has an identity, has a handle, and you can attach concepts or properties to that handle. The surface of the cell doesn't. The surface of the cell is what's called a fiat object in computation. And that means that the surface is not something you can attach information to. And that has its own set of complications. We probably won't run into them too much, but it's something to be aware of. If you wanted, for example, to say that the concentration of a particular channel protein were different in different positions of the cell surface, that requires some gymnastics to implement computationally. On the other hand, the volume of a cell is well defined. You count how many voxels it has. The surface is the number of boundaries between your cell and a different cell, and that number will depend on the interaction range. And so when you change the lattice type from square to hexagonal, the volume doesn't change, but the surface changes. If you go from 2D to 3D, the volume doesn't change, but the surface changes. If you go from nearest neighbor interactions to third neighbor interactions, the volume doesn't change, but the surface does. And that's something we can play with a little bit today. When we want cells to do something, we have to define a set of behaviors and interactions. And those behaviors and interactions are represented in terms of effective energies. That is potential effect, things that have the form of potential energies that generate forces. And the total model, the main part of the model, is the specification of all of the different energy functionals that the cell participates in. In this case, we have an example where we have a contact energy, which we'll talk about later today. That is how much cells stick to each other. Surface area constraint energy and a volume constraint energy, both of which we talked about last time and we'll work with today. Once we've defined the spatial domain of a cell, the energy of the cell, we have to specify what the cell does in response to that energy. And CompuCell uses, and we'll come back to this stochastic dynamics. That is, it probabilistically moves things around in configuration space. That has the disadvantage that it's stochastic, and it has the advantage that it's numerically absolutely stable. So it means that we don't have to worry about simulation instability from that, which is a problem in deterministic methods. Things move around, and we'll say this a second time. In general, they're in the viscous limit or the overdamped limit, perfectly damped limit. That is, if I push on something with a force, the velocity that with which it moves is proportional to the force. So there's no intrinsic acceleration in CompuCell. It's an Aristotelian rather than a Newtonian world. And that is more or less what happens on the length scale of cells in reality. So one thing that came up talking about the homework, and I want to emphasize this, I've said it again, I'll say it yet again, is that essentially nothing exists in a simulation unless you assert it. If you don't say there's a cell lattice, there's no cell lattice. CompuCell will happily run with no cell lattice. If you don't say that there are cell types, there are no cell types except medium, which is a non-cell type. If you want to create a cell, you have to create a cell type before you can create the cell. If you create a cell type and you don't explicitly create a cell, there are no cells. And so you have to do all of these things by hand. Nothing is free. If you create a cell and you don't assign properties to it, it also has no properties. And you can try that and see what happens. So there's no volume for, no target volume for a cell, no target surface for a cell, no adhesion energy for a cell unless you assert it. And that means that cells, once they're created, don't continue to exist or stay spatially connected unless you have some reason for them to do so. And so the definition of these constraints and energies is the critical thing in building a simulation. So we talked about how to do that, and many of the things that we define about cells, like volume and surface, are defined in terms of what we call harmonic or elastic constraints. And the thing to think about when you're thinking about a harmonic or elastic constraint would be a spring. If the spring has an equilibrium length L, and I want to shorten the spring, I have to push the ends of the spring together. And the spring will push back. It will have a restoring force trying to restore itself to its equilibrium length. If I pull it out, to make it longer, it'll pull back with a restoring force, trying to restore it to its equilibrium length. In the case of a spring, it's only pulling in one direction at a time, and so the way the force is generated is pretty straightforward. On the other hand, if I think about a balloon, which is filled with a certain amount of gas, and that balloon is in this room and not moving, then the pressure forces on the balloon and the tension in the membrane of the balloon balance. And so the balloon surface is at equilibrium. If I try to make the balloon bigger by pulling on it, say by reducing the pressure in the room around it, the balloon will stretch, but it'll have a force pulling inward from the expansion of the gas and the membrane tension. If I try to compress the balloon, there'll be a restoring force pushing outward. In the case of a spring, because it's one-dimensional, I always know what direction that force is in. In the case of the balloon surface, the direction of the force is not so obvious. It's going to be normal to the surface, but could be something more complicated. And so in general, the idea of a constraint as something that creates a force that pulls the system to satisfy the constraint is clear. The direction and the magnitude of the force may not be so clear. And one of the nice things about writing things in this energy formalism rather than a force formalism is that the formalism automatically calculates the forces that are required to satisfy the constraint. You don't have to by hand calculate the vector directions of the forces. That's done for you. A harmonic constraint always has the form of lambda. You'll see a lot of lambda volumes, lambda surface, lambda this and lambda that. Is the strength of the constraint. The bigger the lambda, the stiffer the spring. The bigger the lambda, the stiffer the membrane of the balloon. And then some scalar quantity, which is a property which depends on the configuration of the object. For example, the volume of the balloon. And a target number for that value. And you take the difference squared because you want, like the spring, if you push inward, you want the spring to push out. If you pull outward, you want the spring to pull back in. So you want the restoring force to be symmetrical. And quadratic energies are the typical way to do that. You can build other energy forms, but in CompuCell, we'll typically use that quadratic form. We talked a little bit about how you specify models. We'll come back to this. CompuCell static properties are typically specified in CC3D-ML, simple markup language. As I mentioned, something you're going to have in any simulation that has cells is a list of the types of the cells. And there's always going to be a cell type medium, which is empty space in the lattice. And medium doesn't function like a cell, and you can't attach certain kinds of attributes to it. And so if you actually want to do anything, you have to create at least one cell type beside medium. You could have as many different cell types as you want. If you want to specify target volume, this was the mathematical form of the target volume. That maps onto a plugin name. Plugins are energies in CompuCell called volume. And it has a parameter for the cell type it applies to because different cells could have different target volumes. Lambda volume, target volume. And CompuCell will automatically go through all the cells of the system, check what their actual volumes are, calculate the square to the distance from the target for each cell, multiply by the lambda for that cell, sum it up, and that's the total volume constraint energy. The same way for the surface. The thing you have to remember about with surface is that the calculated surface area will depend on the lattice type. And so when you change from square to hexagonal lattice, your target surface probably needs to be adjusted because it'll change the calculation. So let's go back to the homework. And based on the questions I got during the past week, I updated the online version of the homework slightly to try to make it clearer. The first homework problem was looking at the cell lattice with one cell in it and asking a very simple question based on the energy functionals that we had just identified, which was, suppose that there's a single cell here in red, white is representing medium, and the only energy in the system is the volume constraint, and the cell has a target volume of 25 and a lambda volume of 2. And so the first question was, what is the effective energy of the image? Then I added this to try to clarify the original question. The original question was a little bit more open-ended. Is there a single voxel change you could make to the lattice configuration? Because the dynamics is always trying to reduce pattern energy. That would reduce the volume constraint energy. And the third question I added, again, trying to clarify the open-ended original question, was, is there a lattice configuration that has zero energy? And then maybe expanding that question a little bit, suppose the target volume were 15 instead of 25, what would you see? Remember, the volume energy, here there's only one cell, so we're only summing over one object, so we don't need the summation, is lambda volume, which is 2, times V, which is the actual volume of the cell, minus V target. And so we can all look at this and count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. So the actual volume of the cell is 21 voxels. Its target volume is 25. So we have 21 minus 25 is minus 4. Squared is 16 times 2 is 32. So I get the energy of this is 32. Okay. Since the actual volume of the cell is less than the target volume, if I made one of these white voxels red, my actual volume would go from 21 to 22. So my energy would go from 2 times 21 minus 25 squared to 2 times 22 minus 25 squared. That is, it would go down from 32 to 18. If I had exactly 25 red pixels, then I would have 25 minus 25 squared, 0. And so if I had 25 red pixels, I'd have an energy of 0. Question: would it matter what the configuration of the red pixels were, provided I had 25 of them? Somebody, I'll call on somebody if I don't get somebody to answer that. Devin.
Can you repeat the question again?
If I had 25 red pixels, would it matter where the red pixels were in this pattern? Would the energy care? What?
No. No, since we're just looking at the volume constraint.
That's right. So it would mean I could have a checkerboard where I had white, red, white, red, white, red, white, red. As long as I had 25 red pixels, it would still have an energy of 0. And that's not very cell-like in the sense that it means the cell doesn't have to stay connected to itself. If I made the target volume 15 instead of 25, then for this initial configuration, 21 minus 15 is 6. The energy would be 2 times 6 squared, 72. So it'd be higher. And if I wanted to get the energy to 0, I would have to go down to 15 pixels box. So if I have only one energy term, it's pretty simple to identify what's going on. I can look at it and calculate it. Now, as I mentioned, you build up a model by adding energy terms, effective energies, each one of which represents a different attribute or behavior or feature of the cells. And so the dynamics that I've defined will try to simultaneously minimize all of these things together. Actually, what it'll try to do is minimize the sum of all of these effective energies. And that usually is not possible to minimize them all. In other words, there is no configuration that has 0 volume energy and 0 surface air energy and 0 this energy and 0 that energy. And so what will happen is the configuration will change to be some compromise that minimizes the net energy, even though none of them individually is minimized. Again, with this particular dynamics, these constraints create forces on the pattern that automatically push the pattern to change in directions and ways that reduce the energy. And that seems a little bit magical, but then again, physicists' concept of potential energy is a little bit magical. And there was a lot of resistance to it as a concept when it was first introduced. Where is the potential energy? It's not obvious where it is. It's a mathematical property of the way the world works. If I make that lambda term bigger, the effective force on the object is bigger. Since velocity is proportional to force, that means objects move faster when lambdas are bigger. It also means that if I'm having to balance out different terms, if I make the lambda for one energy term bigger, that energy term is satisfied more closely than the one, and the energy terms conflict. The energy terms with big lambdas win, and the energy terms with small lambdas lose. And that's something we can see also in the real world if we try to satisfy 2 constraints simultaneously. I want to get a perfect score on my homework, and I want to get some sleep. Well, I could stay up all night working on the homework to get a better score on the homework, then I don't get the sleep. I could sleep 12 hours and not work on the homework, in which case I do fine on the sleep score, but not so well on the homework score. We have those kind of conflicting constraints on us all the time, and we make some kind of compromise between them. The same thing happens in these simulations. Does that help give some idea of how these things work? It's also true that if you have some lambda or constraint that's huge compared to the other ones, then that one gets satisfied and the other ones more or less get ignored. And so, in principle, if I jump up and down on the Earth, the center of mass of the Earth moves as I jump. But in fact, the center of mass of the Earth moves very little, and I move a lot. And so the Earth's lambda is very large in terms of its center of mass, and so it moves less, and I move more. So that was the point of the second part of the homework problem 1, which was to now look at a pattern where we have both volume and surface, and ask the same question we asked before. And just to make it slightly more interesting, we changed the targets and the lambdas to be slightly different. So here we have both a target and a surface. And let's just say, what's the total effective energy of the image? Well, first I have to calculate the effective energy for the volume constraint. Well, how many voxels do I have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. And its target is 15. So 20 minus 15, 20 minus 15, 5, squared is 25, times 3, 75. So the volume energy of this pattern is 75. What about surface? Surface, again, depends on my interaction range and lattice type. But let's assume we're just nearest neighbor interaction. In that case, it's how many lines are between a red and a white square. Let's start here on this end. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28. Did I miss something, or is it 28? It's easy to miscount. I counted 28.
That's what I had when I solved it too.
So 28 minus 13 leaves me with 15. 15, 28 minus 13, 15. 15 squared, 225. 225 times 3 is 675. So the surface energy here is 675. The volume energy is 75. Surface energy dominates. So if I want to make a single voxel change to reduce the total effective energy, well, both the volume and the surface are big. So I want to get rid of a red voxel, turn it white. But I want to take one out that's going to get rid of as many surfaces units as I can. If I take out this one here, say on the end, I get rid of 1, 2, 3 units of surface and create 1. So my net change is -2 for surface and -1 for volume. And then the last question was, what's the possible configuration to minimize the total effective energy? And is the total energy 0? Why don't you think about that for a second? Is there any configuration of voxels in red that I could draw that have 15, volume of 15, and a surface of 13? It would have been maybe a little easier if I'd had a target volume of 16 rather than 15. But it winds up being more or less the same. If I draw a square that's 16 by 16, 4 by 4, and take out one corner, a 4 by 4 square has area of 16. Take one out, it's 15. Its perimeter is 4 plus 4 plus 4 plus 4. It's also 16. Taking out the corner still leaves it with 16 perimeter. And so if I were to, the other thing I could do is put my lattice into a grid, which was 3 by 5. And in that case, I have two sides of 5 is 10, plus the top and the bottom is 3 plus 3 is 16 as well. So if I want a volume of 15, the smallest surface I can have, perimeter I can have, is 16. So there's no way to make, those will have the smallest energy, but there's no way to make the total energy 0. And so this is doing that calculation again. If I started with this configuration, I got an energy of 750. If I took out that pixel there, it goes down to 555. If I do the best I can, which was to put 15 voxels together in a 3 by 5 rectangle, the minimal possible total energy is 27. That's the best I can do. Okay. Another thing that came up, which I probably, I created this picture, I should probably have created this picture a long time ago, was using Blob Initializer. And there's a pretty detailed instructions for how to use Blob Initializer in the manuals. But I'll go over again how Blob Initializer works. Blob Initializer creates a simple array of cells. It's not very sophisticated, but it's a place you can start. Blob Initializer has the following properties: center, radius, gap, width, and types. And so if I imagine that my lattice is two-dimensional, 0, and it happens that the x dimension of the lattice is called self.dim.x. We'll need that later. The center position in x is given by x center. Center position in y is given by y center. In this case, that we've got a two-dimensional lattice, so z is 0. And there's a little bit of an inconsistency because sometimes the z really should be 1 rather than 0. So it's something funny about that. It doesn't matter, but if you make your lattice 128 by 128 by 2, you run into a problem. There was an inconsistency in the model definition specification. Radius defines a circle around that center position in voxels. So radius here was 51. Gap, we'll talk about in a second. Width is the size of squares. So what Blob Initializer will do is try to fill in a circle with squares, square cells of size width by width. And it will have jaggies around the surface. So it's not going to be perfect in terms of filling in the space. Some of the cells may be slightly small, truncated. Some of the cells may stick out a little bit. So it's an approximate circle. Gap, sometimes I want my cells to be in a circle, but I want to have space between them. Gap lets me define a space, a gap, between the cells. And so here, if I made gap 5 and my cell 7, I'd have centers of 12, every 12 voxels. The Blob Initializer will look in the types list and it will randomly assign the type of the cell based on what's in the list. If I had 2 cell types, mesenchyme and epithelial, it would randomly say the first cell is mesenchyme, the second cell is mesenchyme, third cell is epithelial, and so on. Typically, it picks from this list randomly. So if I wanted 2 times as many mesenchyme cells as epithelial cells, I said mesenchyme, comma, epithelial. I'll have 2/3 mesenchyme and 1/3 epithelial. There's a minor annoyance that you can't have any spaces in your list. So you have to get rid of spaces. It's not English. Okay. If you want to create a single cell using Blob Initializer, the only thing you have to do is make the radius of the circle smaller than the width divided by 2. In that case, there's only room for 1 cell, and it'll draw a single cell. And so that was a little bit of a challenge in that homework problem to do that. Okay. I don't want to go through all of the details of cell dynamics again. In reality, cells move by extending and retracting their cell membranes actively using molecular motors, actin. And we're going to assume that those movements are quasi-random. Whether they really are is a good question. Our goal in the end is that we want the velocity of things to be proportional to the force. It's what's called a Monte Carlo method, which is that the cell will randomly, the system will randomly pick a voxel, and it will ask the question, should I change the value at that voxel or not? And if it does, and then it will evaluate the energy. If the energy is decreased by making the change, you do it. Water rolls downhill. But because there's active movement, sometimes things move uphill as well as downhill, which again is not something you're used to in Newtonian physics. If you do thermodynamics, then you're used to it. There are a couple of details which we're not going to go into very much. I mentioned that voxels could separate from their parent cell, and that's possible, but because you do only nearest neighbor copying or next nearest or third neighbor copying, that's given by that copy range, that interaction range that we define. voxels can't move very far or very fast from the original cell. And that actually has some rather specific properties. If you're a physicist, there's something called detailed balance, which isn't satisfied by that algorithm. And since the world doesn't actually satisfy detailed balance, that's what you want. What you do get, though, is that the probability of going uphill depends on the force you're pushing against and how hard you're pushing. That random force is pushing, and that random pushback is given by this letter T, which has the unfortunate name of temperature in some places, but is really a fluctuation amplitude. And this does have the property that if the changes in energy, effective energy, divided by T are all small compared to 1, that you have linear force-velocity relation. So we had a homework problem, which was, what was the behavior of a cell with only a volume constraint? And I asked you to create a cell with a volume constraint only, and then run it and see what happened. And we did that. And how many people were able to do that? Maybe use the button. Well, maybe it's a different question. Was there anybody who was not able to do it? No embarrassment if you couldn't, but was there anybody who had trouble doing it? Everybody got it okay? I could do a poll and ask. Feel free to speak up. Okay, so what you should have seen is that you created a set of voxels, which represented the cell, but because the only energy in the system was the number of voxels in the cell and nothing to do with the configuration, the voxels could separate, and you could see that the voxels you get these disconnected pieces of cell. Now you could say, why aren't the individual voxels moving away and turning into dust? Why isn't the thing distributing completely? And the answer is that if you wait long enough, that could happen. But this algorithm of updating does local updates. And so it takes time for voxels to move from where they start in a cluster further and further away from their initial location. And so there is some confinement due to the dynamics, the Potts dynamics in this problem. And if you want to try running that, I created a simulation. It's in the online, it's up on Canvas. It's also in the shared Google Drive, just called Cell Volume Demo. And so the key thing to notice when you run the simulation is that the cell volume is more or less conserved, but the voxels migrate apart from each other. The cell falls apart. And the last two problems were, now let's try to fix that by defining the surface area of the cell as well. And then ask the question, if we make the surface area bigger or smaller compared to the volume, what happens? And also potentially if we change the strength, the constraint strength. And perhaps for C and D, we should have said constraint strength change rather than target strength. And so the best way to do this, if you're going to do it as an exercise, you could certainly change those values inside of the code, edit them, intuit it, run it, edit it, intuit it, and run it. But one of the things that you can do conveniently in CompuCell is that you have what's called steering. And steering, if I run CompuCell player, and maybe it's actually better for me to have player running here. So let me get player. And you might want to follow along if you feel like it. And I'm going to use the desktop version of CompuCell at the moment. And you can also use oops. Well, that's one reason not to use. That's one reason to. Maybe I will use. Maybe I will use maybe I will use the. No, shoot. Unfortunately, let me try this one more time. Unfortunately, Zoom and Player and Twit sometimes don't play nice with each other. And so there's a font size issue. If this doesn't work this time, I will try doing it. I'll try doing it with the one online. There's a font size issue with Twit when you're running Zoom. No, sorry about that. No, it's not gonna behave. No. Okay, sorry. Give me a second. I'll launch it on NanoHub. See, if NanoHub will behave, NanoHub seems to not want to behave either today. Okay, is that visible all right?
Yes.
All right. So just to let's run, I'm not going to run the particular simulation I was talking about. I just want to talk about steering for a second. If I open a CC3D project, and I didn't upload these beforehand because I was going to run them on my desktop. Bad mistake. Let's see if we can find the CC3. Okay, that one should be all right. Actually, this is close enough to what I want. Actually, I can actually get this one to do what we need by just uncommenting here. Comment. Okay, so this is the simulation we've been talking about. And now I'm going to run this in player. And I see, actually, it looks like somehow, okay, I've got volume. This is the case, this was the first one where I didn't have the surface any to specify. So if I go here to steering, if I go in this left-hand model editor window and pull down plug-in, volume energy parameters, cell type, cell, lambda volume 2, target volume 50. If I make it 125, I see my cell grows. If I want to change lambda volume, I have to type it as a float. I can't type an integer, but it'll change. And so that steering is something that you can do to explore things without having to stop the simulation and run it again. There are other ways of steering the simulation that we'll come back to later, but that one is a convenient one to have to start. It's worth saying that only the things that were defined in the XML will show up in the steering window. When we start doing Python definitions, as I just said, we'll have to do some additional work to create widgets that will allow you to steer Python values. And if people did it's not an IPy widget, it's a different widget set. But if people did slider-based steering last semester, you'll be familiar with that. One minor inconvenience in CompuCell has been on the stack for things that we were going to change and fix for a long time, is that when you make a change here, like I changed the target volume from 125 to 250, it's not logged. There really should be a run file that keeps track of when those changes are made. So it's important, if you think that it's going to be interesting, to write it down. It seems sort of archaic these days, so that you can find the parameters that you liked. So if we have a simulation where we can change the target volume and surface to meet the relative proportions, then we would want to use that steering option. And let me see if I can actually get this to work. In my Twidit, I have the surface energy defined. Let me see about saving this. As a new file. Let's call it single cell volume surface. Okay. And now let's open that in player. Let's see if it works. It's taking its time. I'll get it. So this one, let's see if this works now. Notice that here, before I only had the volume, now I have the surface as well. And now, if I look at my cell, it's much more compact. It still has some pixels coming off, but not as many. And so if I now make the target volume big and keep the surface the same, that's like blowing air into a beach ball. And the cell gets stiffer and rounder. if I blow enough air into the beach ball by making the target volume very large, that I will stretch the membrane of the beach ball, but I'll get less and less fluctuation. It'll become more like a basketball or a golf ball. If I make lambda volume bigger, that means that the volume constraint is more important than the surface area constraints. If I make it go from 2 to 5, well, it didn't do much. I could play with that a little bit more. If I do the surface constraint, it's got a target surface of 50, target volume of 500. 500 would be what? What's the square root of 500? So we make it something that has, let's pick a number that has a square root that we know. How about 900? That would be 30 by 30. We make the target volume 900, 30 by 30. The smallest surface we could have would be 30 times 4 would be 120. Here, the target surface is much less than that. So our cell is very compressed. If I make the target surface of 120, I should get something that's more or less round. If I give myself more surface than that, 240, I'm trying to create extra surface for that given volume, and the surface will get dispersed. If I make the target surface very large, 2000, it will try to create area by having it fall apart. So did people see something like that when they tried doing this simulation? So this, I hope, gives you some idea of what these energy functionals can do by themselves and together. You'll notice that pixels are coming in and out of existence here. This doesn't look like a real cell at all. And yet, when I picked the parameters correctly, it behaved a lot like a cell. It looked like a cell exploring in space and crawling around. So if I go back to 120 here, well, the thing, sorry, I had 900, so 30 by 30. One thing that you'll find is if you change these things too fast, things tend to freeze up. Or else I had 2000. And so I may want to stop it and start it again. If the configuration that I'm in is too far from equilibrium, it can't find it. Okay, so here's a cell. It's not completely simply connected, but it actually is not doing a bad impression of an amoeba trying to explore. And you'll notice the center of mass of the cell is moving around. The cell is doing a random walk in space, which cells really do. So now we can understand what high target volume versus low target volume mean. If the target volume, if the minimal perimeter of the cell is a lot bigger than the perimeter for a cell of a cube of that volume or spherical of that volume, then I have a high target surface. If the volume is very large compared to the perimeter, then I have a high target volume. And again, the thing to think about would be a beach ball. The target volume is the amount of air I put in the beach ball. The target surface is the size of the beach ball itself. That is the rubber of the beach ball. And I can play with that. This particular simulation is uploaded. The stochastic dynamics picks lattice at random and tries to change them. That defines a conceptual time, which is trying to update each lattice site in the simulation once. Now, in practice, only lattice sites that are near my cell do anything. And so if I pick a lattice site way over here in the corner, I'm wasting voxels. And there's actually something called Box Watcher, which draws a rectangle around all the non-empty voxels and only does updates inside of that region. It speeds up the simulation a lot if we were doing this kind of simulation. The next thing you'll see is there's something called run Steppables. So one Monte Carlo step consists of trying to update as many sites as there are sites in the lattice. And he often gets written as updating every site in the lattice. But because I picked places randomly, it's possible that some sites get picked more than once and some sites don't get picked at all. So if I'm, say, on a 256 by 256 cube lattice, or let's say 100 by 100 lattice, I have 10,000 sites. 10,000 attempts to move the boundaries is one Monte Carlo step. But it's possible some sites get picked once, more than once, and some sites don't get picked at all. One could, in principle, take a list of all the sites and do a shuffle instead of picking randomly. And that would have slightly different dynamics for the system. Actually be an interesting thing to investigate. These kind of Metropolis dynamics methods have been studied a lot over the years. As I mentioned, they were originally developed in the context of the atom bomb. And so there was a lot of work done on how they work. This particular updating algorithm is one I developed. It wasn't a standard one. And it's appropriate for the particular system. In any case, one Monte Carlo step is one sweep through these lattice updates, and then CompuCell will look at its Python code called Steppables and do whatever you've done there. And they're called Steppables because they're run by default once per Monte Carlo step, although in principle you could have them run every 5 Monte Carlo steps or every 10 Monte Carlo steps. It would be convenient if you could run them more than once per Monte Carlo step, but that's not an option at the moment in CompuCell. There's no reason it shouldn't be, but it is. All right. So now I'd like to turn the floor over to you just for a few minutes and ask people. I know it was sort of a late assignment, and if you didn't do it, that's okay. But I asked you to maybe just do one slide or a quick comment on project ideas, because I was hoping that people could potentially begin to see, again, people are welcome to do projects on their own, but I was hoping that if there were people who had shared project ideas, you could begin to coalesce around a project. And so I wanted to sweep the class and ask the question, how are people going along with project ideas? Next week, I'll ask you to do a more formal presentation of a project proposal. But for this week, it was informal. And so let me turn it over to you and ask you, just going around the room, if you've made any progress on thinking of a project, and if so, if there's a shareable, a shared interest among multiple people. So maybe start with Fanbo. Did you have any ideas that you've developed for?
Let me share screen. Okay, so basically what I'm interested in doing this project is to apply the Mechanica to the NanoHub application. So because I have previous experience in doing the Python Jupyter Notebook GUI in turning the C++ code into the the GUI in NanoHub, and this window is an example of the Nano confinement app in the NanoHub application. So I was hoping to doing this Python-based project, but right now I don't have any concrete ideas of what application should be made in like this, using the Mechanica to do something like to model like a cell sorting problem or some other problem. So I would like to see if anyone has like some ideas, and I would of course like to welcome to join like the group if
Right.
you are still like interested in like applying the Mechanica in the Python.
Well, I think what you've done is really good because Mechanica is fundamentally a molecular dynamics simulator.
Yes.
And so since you've been working with a molecular dynamics type simulation, that sounds wonderful. And so potentially if other people also want to work on Mechanica projects, that would be fine. And you could come up with a joint project. Again, there'll be some disconnect then between the lectures and the homeworks and the project, because the lectures and the homeworks will be primarily focused on CompuCell. But we can try to open things up. In other words, we could ask the question. One of the things that I wouldn't necessarily demand from a course like this, but it's actually quite important from the point of view of using these simulations in medical applications, for example, is that you actually would like to have some ability to check that the simulations you're doing are correct. And one way of doing that is to try to run a simulation of the same thing using different methods. And so it would be quite interesting, for example, to write a simulation in CompuCell, which uses this lattice-based method that we've been presenting and has these extended cell objects, and write a simulation in Mechanica of the same thing using a center model approach off lattice. And if the biology and the physics are the same, you should get the same result. And so it would be nice to actually write a simulation, say, I mean, Andy's been working a lot on these simulations of zebrafish apibole, and to see if you could compare the two results. Again, I'm not saying you have to do that within the context of the course, but it could be quite interesting.
You know, I mean, what I was thinking for a while that might be a really good example is Josh's that paper that Josh worked on, where he started with a subcellular model and then ported it to use POTS, and then we could implement that same subcellular type cell division type model in Mechanical rather easily. And then and considering that Josh has the CompuCell version of it, and I think that would actually make a pretty interesting paper of actually showing both simulations side by side.
Well, certainly being off lattice should do well in that kind of simulation. You're going to have subcellular element type cells, though. So let's have an offline meeting on that. I think that's not a bad idea. I mean, another kind of thing would be the cyanobacteria or bacterial swarming simulations, or some of these. Again, it depends a little bit on what pieces of Mechanica you have, because you don't want people to be waiting for you to write new core code.
Yeah, well, I mean,
it has to be something that's achievable now.
Yes, I mean, so ideas like the cyanobacteria would be good. The subcellular element one would be good because all of those are working right now. And if you're interested in cyanobacteria, I have a researcher in UK who I've been talking with, who's interested in writing a paper on that subject.
Well, certainly the kind of motion that they have is it's possible to do it in CompuCell, and people have done it. Andreas Deutsch has done it with his POTS simulator, but it's a little more difficult to do it because of the scale issues. So good. So let's hear what other people have to say, but I have no problem with that as a possibility. So let's see who's next on the screen. Kali.
All right, so I have three main project ideas at this point. The first is cancer therapies, pretty straightforward, simulating the introduction of a drug therapy to a tumor, likely expanding on one of the provided papers listed under cancer therapy, perhaps looking at a different type of cancer than the ones that have been previously explored, or maybe trying to implement a non-chemotherapy based treatment. From what I'd seen, all the papers, I think, were pretty focused on chemo. So there's some ideas to maybe introduce oncolytic viruses as like a chemical species in CompuCell, or somehow incorporate immuno-oncology into all this. Not 100% sure how that would work, but I feel like there might be a way to do it.
Well, you certainly could have a representation of natural killer cells that come in and attack the target cancer cell, or CD8 plus T cells, some other T cell species. And then you could in principle have a model of immune hiding where the tumor secretes some molecule that at least some cells in the tumor secrete a molecule that inhibits that killing. The one thing you face always with those kinds of simulations is essentially simulation size. If you're in principle, these simulations can be very large, but in practice for a semester, you don't want to have simulations that take weeks to run on the cluster.
Right.
If you're doing it for production code, you throw 10,000 jobs on the cluster, and if they take 2 weeks, at the end of 2 weeks, you've got 10,000 jobs. But if you have to have a result within 13 weeks, then typically you run smaller simulations to get the ideas across. But I think those are both quite quite possible. How ambitious you get is another question, but those are both realistic. And Josh has done cancer simulations. So, I mean, of course, you've done them with Paul, so you know quite well the biological concept. But Josh has implemented cancer simulations inside of CompuCell, so he has experience doing that. And I also have done cancer simulations, so that's certainly possible. Okay.
Okay, second idea, similar, putting a therapy, but this time on a biofilm. So this would expand on the work by Poplawski et al.
Yeah.
So that's also just pretty straightforward in the idea there. The third idea is actually one that I got from my cell biology professor. I asked what would be some interesting multicellular system to model, and she gave the idea to maybe try to model cell signaling. So I was thinking we could introduce some amount of a cell signaling molecule into a multicellular system and then simulate the propagation of that signal in the system. So you would have two types of cells, those which have received the signals and those which have not. Where it gets complicated is if I would potentially track the concentration of some sort of resulting compound in the signaling pathway. And a lot of the times those compounds have feedback inhibition on different parts of the process.
Right.
So I feel like that might be a little complicated to look at, but maybe I don't have to go that deep. I don't know.
Well, again, I think that one is very similar to the work that was done by Stan Murray and Nick Saville almost 20 years ago now, where they on Dictyostelium discoideum. So Dictyostelium are amoeba. When they have plenty of food, they live on their own and they munch on bacteria. When they starve, they send out a cyclic AMP signal. And sort of counterintuitively, when one amoeba says, "I'm starving," everybody rushes to join it. I would think you'd want to go the opposite direction, because if there's a signal saying there's no food where I am, you'd think you'd want to leave rather than occur. But they flow together, and that flow is there are two things that make it interesting. Well, there are a lot of things that make it interesting. First, the relaying of the signal is pulsatile. So the cell sees a rising concentration of cyclic AMP. It responds by releasing a puff of cyclic AMP into its environment, moving in the direction of the source of cyclic AMP, and then it stops for a while. So there's a refractory period during which it can't release cyclic AMP anymore and it doesn't move anymore. So basically, it sees a puff of cyclic AMP, it sends out a puff, it takes a step, and then it stops for some amount of time. And then it becomes activated again or excitable again and does the same thing. And those simulations are not implemented in CompuCell. They predate CompuCell, but they're quite pretty. It makes some beautiful, beautiful movies. I've wanted for many years to have somebody re-implement those old simulations of Saville and Murray. The one thing that can be a little bit tricky is that I think there's some typos in some of those papers. And so the actual parameter values that they published may not quite be the parameter values they used. But at least in principle, those are doable. And that would be, one could build could build it. So you could start out by having the cells not move and do relaying, and you'll get traveling wave patterns and spiral waves. And then you add the movement of the cells in response to the signal, and you'll see this streaming behavior that you get in Dicty. And that's certainly a practical thing to do. It uses almost everything CompuCell has, and it might require you to jump a little bit ahead because we may not have time in lecture to get to chemotaxis and diffusion in detail until week 6, and you might want to start before that. But that's a very achievable. This kind of question is very achievable. If you include movement, it's more fun. If you don't want to include movement, there are developmental problems like coloration in zebrafish, where there are mutual inhibitory signals sent between the chromophores in the skins of fish that give rise to stripe patterns. And there's been some nice modeling of those in other modeling systems. And those I did some implementation of those a long time ago. Not enough to ever publish it, but I did write them out. And so that's also achievable. So all three of these are great projects. They're all definitely doable. So the Dicty papers are particularly, to me, aesthetically, they're very beautiful. The movies that Stan did at his thesis, I was on his PhD committee. I think I said are really some of the most beautiful movies I've seen of this kind of simulation. So we could look at some of that material.
Okay.
So they're all good. They're all the hard thing will be picking one because they're all great projects.
All right. Thank you, Dr. Glazier.
Tyler, I think you're next on our.
Yeah, I can go ahead and share the screen. So I wanted to do the immune response to viral infection, building on the project I did last semester. And some of my initial ideas were maybe I could experiment the length of the duration of an infection based on the viral load or how prepared the immune system is for an infection to occur. I could also integrate, similar to what Callie was thinking with cell signaling, the pathways that the immune system takes to fight an infection. And I had a little uncertainty with if I should incorporate the innate immune system as well. Since this will mostly be in host, I'm not sure how I would do that if I want to think about that right now. But that's pretty much most of the ideas I put together for the project.
So, Josh, you probably have something to say to that.
What do you have in mind?
Well, I mean, you're doing modeling of interferon response. You've done modeling of
Mhm.
NK cell response. Julia,
Right.
I mean, TJ is doing models of CD8 plus and APC cells. So
Right.
so I think all of those things are very achievable, and the question then would be essentially, the issue there is essentially, do you want to start from scratch, or do you want to integrate into an existing project and then figuring out how to do that?
Right, and
So,
especially in your case, Tyler, since you already have a project that you've been working with last semester, is I actually like. I mean, the details. I don't exactly remember, but I do remember. I do think that that is one project that you can continue on developing by specializing the model, and then that allows you to do two things. Allows you to gain some familiarity with CompuCell, but also um, familiarity with the method that we've been developed in terms of how to translate one method to the other, and then be able to compare results from one to the other.
so I think we should hook you up probably
And
with
that
TJ.
is achievable.
Yeah, we could talk to. I think so. We should probably make a note, Josh, that we should hook up with TJ and have that conversation.
Right.
I mean,
But I
that's
think
how.
because what I want to say is that specializing the model requires certain mathematical skills, but it's also important to have an idea of what the mechanistic hypotheses are, the biology is, because that dictates how do you interpret the OD model and how do you move forward in specializing it. So it does require you to have a good understanding of what the biology is and what the model is, and how do you put it in either context.
So I think for that kind of problem, the thing to do is to say, okay, I picked. Suppose you took the one you did last semester. Say, okay, I have this existing model that came from this paper, and now let's think about what really is happening spatially in the tissue that's infected, and how can we model that? I mean, in principle, putting in cell types and giving them behaviors is easy. The thing that's trickier is to try to calibrate things so that they behave properly, so that your CD8 T cells kill cells at the right rate, that they migrate at the right rate, that your APC cells do what they're supposed to do. And so the question for all of those things then become data sources. Now, if you're building an abstract model where you want to say, if you have these kinds of behaviors, what happens? Then it's not. Then you could do that. If you have a particular quantitative model that exists already, PJ has got, and Josh have a lot of experience of taking those quantitative models and translating them into spatial models. But as Josh says, then the key question is, what's the additional question you're going to ask? What is it that you gain by going into space? And so we should talk about which particular illness or virus, for example, we're interested in. And again, there's a lot to do. I mean, the immune system is very complex. And perhaps, I mean, one of the areas may be that with viruses, you don't get this so much, but with bacteria, you often get cysts where the bacteria persist, but they're surrounded by immune cells. Tuberculosis is the classic example of that, where you almost never clear it. You always have it in your lungs, but they exist in equilibrium with the immune system. And so then you get spatial patterning of the immune system and the immune response. And there's been very nice work done by, for example, Denise Kirshner on that. And so definitely those kind of models are quite interesting. And absolutely, I mean, these days with COVID, things like interferon production are quite interesting. I mean, Josh, depends a little bit on what you're planning to do, but potentially these co-infection models could be interesting. So let's, if that's what you want to work on, let's have a meeting with you, me, Josh, TJ, and try to narrow down a little more specifically what kinds of questions would be good.
Okay.
Because we could do, the problem here is I sometimes will say to Andy when he'll say you could do anything, and that's usually not a very helpful answer, because you don't want to do anything. You want to do one thing, and what you want is help deciding what that one thing is. And to be told that you have no constraints makes life very hard. And so I think that the conceptually this is a great set of ideas, and the question will be how to. So, for example, if there was somebody else in the room also to work on this and had a specific disease and a specific system, then pairing you up might be interesting. So, but they're good concepts. Everybody has great ideas. Let's see, where are we? Kali, Fadbo, Tyler, Devin.
Yep. Let me just get set up really quick. Okay. So my project ideas are not super sketched out yet, but just based on reading some of the papers, the stuff that interested me was the cancer progression and somatic evolution of cancers. So cancer progression is basically just the normal life cycle of the cancer and how it interacts with other cancer cells. So I want to replicate those interactions within the cells and then look at signaling events, cell division and growth, and tissue remodeling, along with some other factors. And then somatic evolution, which is basically how the tumors respond to their microenvironment in whatever localization they're in. So some examples of that might be oxygen levels and blood supply of the tumor. And I pulled up a couple of papers that already have CompuCell models for them. One of them is actually Professor Glazier's. And so I might look into those a little bit more. And then if I am going to do one of those, since they're already sketched out projects, I'd have to expand on them. So some ideas I had for that was look at different growth parameters for different types of cancers. For example, brain cancers versus blood cancers or something like that. And then maybe implement some treatment factors. And then also look at cancer growth in a confined environment versus unconstrained growth.
Yeah, I think there are a lot of great questions there. That cancer evolution paper was sort of a frustrating one because it took a very, very long time to get finished. And by the time it got finished, everybody had moved on. And so it was intended to be a sort of a bootstrapping paper. That first one, the emergent stratification paper, was intended to be a bootstrapping paper just to show how you could do the thing. And then it was never meant to be the end. We really wanted to continue that work. The simulation sizes were very small, so the number of cells was very few. The variety of situations that were investigated was very few. The hypotheses about what caused the cells to die and survive was not very well implemented, and so that's a paper that could definitely be redone with more modern technology to be much better. Maciek would probably be very happy to talk to you about that. Even though Maciek's still not here in IU anymore, he still works on CompuCell. He's still interested in that problem. As I say, Josh has worked on cancer modeling, so he might be able to help out. I could certainly help out. That second paper, we don't use the Bionet solver. We use the Tellurium to implement the pathways, but you could definitely do that. Mark, I don't know how available Mark is at St. Andrews, but Mark was Paul Macklin's boss for a while, as a matter of fact, in the old days. So Mark is at St. Andrews in Scotland. But yes, those are again good projects. And again, if there were two people who wanted to work on a cancer project that we could come up with an agreement, that's fine. Again, everybody can go off and do their own thing. There's no rule that says we can't. But these projects tend to be big enough that working in teams is also not a crazy thing to do. So, okay. Let's see. Drew.
Hi. I'll admit I didn't see the email, but I did throw up a quick slide here.
That's okay. And sort of last minute.
All right. Is it showing?
Mm-hmm.
All right, cool. So I've got like 3 sort of different project ideas, and they're all not exactly specific, but for the first one that I think is pretty interesting could be just looking at agent-based systems. I read the abstract for this paper here, and it was sort of using like fitness functions to kind of highlight like the most important features to a specific situation in cell growth that was in that type of tissue there. And I thought that was kind of interesting. I might want to do something related to that and observe some sort of patterns that form in some simulation. I don't know. But for my second idea was drug delivery, which I saw Callie was looking about going into. So maybe we could collaborate on something like that. I didn't really get too specific in this one because I haven't read as much on this, but I don't know, that was something I was interested in.
Actually, I'm embarrassed to say I don't remember that paper. It's I definitely, we could look back at that, and I'd be happy to look at that one and see what's in it. I mean, it's clearly a CompuCell paper because it's got Machek and E on it, but I don't remember what it was. But yes, I mean, if there's a shared interest in cancer, and people want to work together on a project, that would be fine. The first one, I'm not sure. Maybe for next week you could show us some pictures from the paper and explain it a little bit more, so that we could try to understand that a little better. As I say, I realized it was sort of a last minute thing to ask you to do something this week, but I thought, again, since the semester is so short, I wanted to push ahead to get people's ideas. And again, I think we could probably rope Machek into at least having some conversations. I don't know how much time he has, but if nothing else, he could explain his papers. And we do talk to him on a weekly or every other week basis. So those are both good. This issue of drug delivery is interesting in the sense that in cancer, one of the problems with chemotherapies is that they don't get into the tumor uniformly. And also that the quiescent cells are quite resistant to being killed. And so what happens with a lot of chemotherapies is you kill the rapidly replicating cells at the surface of the tumor. And then the cells that were quiescent inside the tumor then get plenty of nutrient. They go back into the cell cycle, and a few weeks later, you're back where you started. That's one of the reasons why somatic evolution in tumors is so fast. And so those are both good problems. I don't remember that what the the drug delivery question was for disseminated cancer. There's some very nice spatial models of cancer that were done by the Moffitt people. I'm trying to think, blocking on his name now. He was modeling metastatic prostate cancer in bone. And it's a model where they have, it includes essentially the hypothesis for why you get prostate cancer metastasis to bone, it's so lethal, was that it happens that the cell signals in bone that exist to maintain homeostasis, the growth factors, the prostate cancer cells respond to those growth factors, and they also send out growth factors that stimulate the production of more growth factors that they like. And so that was a model that was done using a cellular automaton method originally. And it might be interesting to see if it could be implemented in a more spatially detailed way. So that's another possibility. That sorry, I'm not. I could if we talk separately. I can pull up the paper. I don't have it at the top of my head at the moment. But that's great. So if there again, people don't have to work together, but if there's a consensus on cancer-related or other problems, I'm happy. All right, I guess we have Jeffrey. Jeffrey.
Yeah, 1 second. I didn't have time to make slides, but I have some papers here. So I mentioned I was interested in fluid flow type of stuff, and I found these papers here. I've been looking into PhD programs to apply for this fall, so this is a paper from someone I've been interested in looking at applying to. So the idea here is that there's this little section here of an enzyme, and these spheres have chemicals that diffuse out of them and react with it. So the product of the reaction is less dense than the reactants. So what happens is the product will float up to the top and create a fluid flow that you can kind of see down here, where this square, right, where the reaction takes place. And you can see that the concentration of the particles moves inward here. So this is kind of the direction I've been going into right now. I've just been reading a bunch of papers on it. So yeah, I don't have a firm idea right now, but in another paper, I think this one, yeah, there are applications to like drug delivery and moving cells around, because you don't need yeah, the flow can move what they call passive particles as active particles. So yeah, this is the
I feel slightly guilty here, because last semester also that the single thing that CompuCell is probably least good at is advective flow. It was designed basically to model solid tissues, and doing convection. You can do it in CompuCell, but it's very, very computationally expensive. Modeling Navier-Stokes is an expensive computation. Now it's not that you can't do it, but it's a computationally inefficient way to do it. And so for the particular simulations that you're looking at, replicating those in CompuCell, you could do it, but you'd be better off using Phoenix, which is code that's designed to do hydrodynamic simulations. I mean, ironically, doing your coupled neural network oscillators, that's something that would be easy to do in CompuCell. And so now, I mean, it's in principle. Andy's wanting to speak, and so in principle, Mechanica can do this kind of fluid calculation that you're talking about. It actually would be much better for doing it than CompuCell would be. My concern is that there are pieces of what you would need that Andy hasn't implemented yet.
Yeah.
And so you would then be totally dependent on Andy getting that work, those extra pieces of code done to finish your project. And so, I mean, that's between you and Andy to some extent, but that's up to you. But you would be putting yourself slightly at risk in terms of finishing up, because if there were a piece of the code that wasn't written, you couldn't finish. And some of what you need is implemented, but not all of it. But Andy, you could speak to that. I know you want to.
I'm thinking, I think we're, I mean, we would need to go through all of the processes involved here, but I think, I mean, in terms of particles where they carry stuff along with them and react with other objects, I mean, that's exactly what Mechanica is designed to do.
Right, but you don't have a viscosity, true viscosity yet, do you?
True viscosity?
You don't have viscous dissipation implemented, do you?
We have a friction force that acts on individual objects.
Well, there'd be a lot of validation. You'd have to show that you get the fluid. You have to show that you get all the right Navier-Stokes behaviors.
Yeah, I mean, we have a.
And let's. But so again, I'm not. So my feeling for this one is, if that's the direction you want to go, boy, maybe Andy should be teaching the course on Mechanica. I should watch and learn. I would say it's possible, and it's the person you're interested in working with is what, Anna Boss? Is that is she the one who's
Henry Schum.
okay? Okay. Because Anna, I know. Henry Schum, I don't know. We work there at Pitt. Was it Pitt or Carnegie Mellon?
Anna is at Pitt and Henry is at Waterloo in Canada.
Ah, okay, that's fine. Because the Pitt people I know pretty well.
Yeah.
Okay, let's schedule a time to meet and talk about it, and pull Andy in and see. I mean, diffusion in CompuCell is fine. Some kind of chemotactic response is fine. You can do complex fluid flow in CompuCell. But it's very slow.
Okay.
And so, I mean, if you wanted to do a, say, 2D section with recirculation, we could do it.
But you couldn't recover Navier-Stokes with
Yes,
POP.
you can. Yes, you can. We've done it. We published it. 15 years ago.
With inertia.
Yes. So you can do it. But it is expensive, and so the simulations are slow, and they're methods that are probably more appropriate. The time you do it is if you want cells plus fluid, then it's worth the cost of doing it.
Okay.
But we could come back to that, and I'm not averse to it. And again, I'm not averse to people working with Andy on Mechanica. It's, as I say, the only thing is that the lectures are going to be a little bit a different direction, but the concepts aren't going to be that different.
Yeah, I was
So
hoping to do a more neuroscience related project for the class with John Biggs, so I was hoping to go in a little bit different direction for this class.
that's fine. That's fine. I mean, I want to support people as much as I can, but I also want to be honest about the limitations of the tools. I mean, something we did before is you could define. I mean, the problem is we're going to run into the same issue I'm a little worried about with Andy, which is that I don't think that the fluid solvers in CompuCell, we have to check with Botcheck to see if they still work. It's been so long since we did the Navier-Stokes solutions in CompuCell because nobody used it, that I don't know if they're still supported.
Okay.
It may have been something that dropped out of the release because nobody was using it. But there are some very interesting things we could do. And CompuCell originally was designed to do the predecessors of CompuCell were not designed to do biological cells. They were designed to do complex fluids, going back far enough. And there were we did some papers on that back in about 2005, 2006. And so let's meet and talk about it some more and put Andy on the call too and see what the possibilities are.
Yeah.
I'm fine with it. Okay, I guess it's one person. It's Furkan. You're the last on the call.
Hi.
Is that right? Have I gotten through everybody else?
Yeah. So actually, I like the idea of epibole in zebrafish, and I want to just explanation what is epibole. Epibole is the last step of gastrulation, and where the cells are kind of like crawling around the yolk, and there are some layers, and in the zebrafish it's a tripoblastic, which means that there are 3 germ layers, and as you can see in here, like there's an envelope layer and yolk syncytial layer and deep cells, and there are some molecular mechanisms that controls those kinds of movements, and for instance, there are E-cadherin is an important molecule for that one, and also there are some signaling networks that is having a role in this epibole, which could be like WNT and other like EPH or JAK-STAT signaling or MAP kinase cascade. So maybe like adding those kinds of interaction to the like model, it might be a good. So I actually downloaded and have a chance to simulate the Epiboli example in there. So and you can see those enveloping layers are actually moving through the yolk cell, like which is a just yolk particle. And also I have other ideas, for instance, like maybe just continuing the previous model, adding more features to it, or maybe that might be an interesting one, reproducing the same model with different benchmarks like CompuCell 3D. And also I know of Morpheus, I can use Morpheus too, and Mechanica and Physicell in the same same domain, and to do again like zebrafish epiboli might be one of them, and two species biofilm or intestinal crypt. Or another idea, I can work on a technology development in the Mechanica's roadmap too. Yeah.
I think those are all good. I mean, I think Andy would love to have somebody with your biological expertise to work on the epiboli. And I think I mean, since you're a Physicell user, Mechanica, right? Mechanica is also a center model, so in a sense, the issues with modeling are going to be similar. If you're used to Physicell, the model specification will be quite different, but the issues about what it is you represent, how you represent it, will be very familiar. And so that could be quite interesting. I think the idea of trying to do a comparison study is also very interesting. Again, the Piboli is particularly nice to me from a Mechanica point of view because you're dealing with these large-scale flows of tissue. That is, as I said, for poor Jeffrey, when you have large-scale bulk flows, then these lattice-based methods aren't as efficient. They work, but they're slow. When you go off lattice, you don't have the issue that you're dragging against the lattice, and therefore these bulk rearrangements are easier to handle. And so I mean that original colonic crypt model you did was very nice. I think you could go a long way with that. And you could certainly re-implement that inside of another framework, whether it's Physicell or Mechanica. The epiboli is really a beautiful problem, and a good model of epiboli, I think, would be very, very nice. It would be challenging, but it would be really nice. The issue for me with epiboli again comes back to what Andy could get implemented fast enough. You might wind up having to implement some core code to get that to work, and so that would be more demanding. But we could sit down and look at what we should probably do if there's going to be a sort of a groundswell of at least some people wanting to do epiboli, would be to look at what we know about the signaling networks. Now, our collaborators at Purdue, Lynn and David Umalis, have done some reaction diffusion models of epiboli. And so they have some numerically validated from experiment models of some of the signaling. And so the problem at the moment is that Mechanica doesn't support the things you need to do that. And so the question then is how much work it would be to implement that. And again, Andy is going to say, oh, you can do it overnight, which he probably could do. But you'd be but you don't want to. Andy also has other things to do. So the problem is whether he could do it overnight, whether it does get done overnight, are different questions. And so to me, the epiboli, there's sort of a catch-22. CompuCell has the support for the signaling, as you know from your previous work, but it's not as good as at these continuous bulk flow as an off-lattice method. And so we should think about what we could define. I think it could be a great project, though, and I'd be happy to see it again. From the point of view of the lectures, I'm not sure everybody will need a different set of lectures from what I can teach, but I can sit and learn too. It's all right. But then we'll have to figure out a way to sort of divide time for class time between projects, because for people who haven't done the multi-cell modeling before, I want to make sure that they get the basics. But that's great. So it sounds like there's sort of a group of people who are interested in Mechanica projects involving bulk flow, whether it's a non-biological system like Jeffrey's or biological flow like the one like the Pimbly. There was some interest in biofilms. Again, Mechanica would be quite good at doing biofilms, although you need the diffusion solvers. That's the problem. But CompuCell's we've done a lot of biofilm work with CompuCell, so we know how to do that. So I think let's try to have some meetings, smaller meetings, to keep developing these ideas. I mean, the only one I'm a little worried about is Jeffrey's project, not because it's not a really good project, but just because I'm thinking about sort of how many steps it is to deliver it, and I don't want people to be too frustrated, and so that's but that's great. So there seems to me to be some consensus about some possible project. It sounds like a cancer. There are definitely a group of people who are interested in cancer modeling, and exactly what direction that goes, whether it's a somatic evolution model, a drug dosing model, an immune therapy model. One could combine those. That sounds like a potential team. And then a Mechanica team possibly doing Fadbo working on the software side of it, and Furkan working more on the model implementation side. That might work. And Jeffrey, I know that the Pimbly sounds very different from the advection problem that you're looking at, but in fact, it's solving almost the identical set of equations. It's just in a slightly different geometry.
Yeah. Actually, it is the same equation. It's just the functional form of the force is slightly different between a free particle versus a bound one.
Great. So good. Well, let's try to get set up some meetings in the next few days. Again, you've heard each other, so I'll ask you to self-organize and try to communicate between you to see if you could talk maybe independently a little bit about it. And if people work as teams, that's wonderful. If you work on your own, it's fine. And if people need guidance on digging up old papers or possible papers, like if you want to do the dictyostelium simulations, I can point your way to that. That's great. Shall we take a? We can have a more general conversation. I mean, everybody has the mic, so if people want to talk to each other now, that's fine. How do people feel? How do people feel about this range of ideas and projects? I think it's. If even one or two of those happened, I'd be delighted. If they all happened, I'd be ecstatic. And when I say that, what I'm saying it, I'm not saying you're not capable individually of delivering them, but it's a lot of science that's covered in those proposals. So please open the floor to people's conversation. I mean, if nobody has anything to say, that's okay too. But you're welcome.
What I will say with the signaling ones is we are designed to have Actually, we already have the concept of a state vector, which is a vector of species attached to each object, and which is working well. And it's working well where you can define fluxes between objects to do diffusion, that sort of stuff. To do the time evolution of the local reaction term, that's obviously something that what is a core goal. I was sort of planning on holding off on that until a little later in the year, but if there is a big demand, it would be from a software side. And I know I'm using this word. It's a matter of hooking up the existing Roadrunner engine with the state vector pointer on each object, and internally on my side, basically writing a little bit of glue code of throwing an SBML, add Roadrunner, having it run in there. It performance-wise, it won't be terribly efficient because we will have n copies of it running. So performance-wise, it won't be any worse than. It won't be terribly ideal from a performance standpoint, but it's possible in a relatively short time frame to throw something together and hook up Roadrunner as it is to, I mean, literally all it is copying a memory block from one to another and calling the Roadrunner step in each time step of the main simulator loop.
I think that would be fine. Again, I mean, if you have diffusion, that then you can do quite a bit. You for viscosity, I guess you have to think about how drag works. And so if you were going to try to do what Jeffrey's doing, but again, let's save that. Let's have that call. Let's have all the Mechanica people on a call.
Yeah.
With you and me. And schedule that for Thursday or Friday if we can.
Yeah, but what I will say, look in terms of viscosity, I have an extremely simplistic concept of friction / viscosity now. To do true viscosity would require a little bit of changes for me. I mean, it's something that I was going to hold off until a later date, because
I mean, if we were a year from now, Mechanica would be ready, and then none of these problems would be happening. I'd probably be teaching the course of Mechanica. But we're, you're working with Mechanica is a great opportunity because it's new, but it's also a little bit of a risk. And so you as students have to decide what your risk benefit profile is. I can't make that decision for you, and I can't teach it because it doesn't exist yet. Andy can teach what he's done. And I know he'd like to. I know he's looking for people. But again, the lecture side of the course is going to stay with CompuCell because that's what's done. But that doesn't mean that you can't work on the other side. Great. So let's try to get some meetings on the books. Andy, I'll let you do some scheduling. And Jeffrey, I mean, why don't you and Andy have a serious conversation about what would be involved in at least implementing convection, thermal convection simulation.
Yeah, I can do that.
And see if that's practical. And the same thing for kind in terms of you have some particular set of signaling pathways. I mean, that if you could write the pathways at SBML, then Roadrunner can solve them, right? You write them in antimony, implementing them in CompuCell or other systems will be the same. So I think those are practical. And then for the people who haven't done, for the people who all of that is gobbledygook, that's all right. We'll keep working on the things that we're working on. And I think everybody has a project that's deliverable. And we have definitely have expertise to help you with all of Okay, do people want to take a, we're running a little behind in terms of where I was planning on being, but I think it's more important to talk about these things. Do people want to take a 5-minute break at this point? Yes, no? All right, well, I'll say we'll take a 5-minute break and then we'll come back to some CompuCell basics in 5 minutes, okay? Okay. And again, for people who, for whom what was just said was not meaningful, don't worry. The expectations of the course are that you learn something, and the lectures will keep going on the assumption that you haven't done this material before. So don't be intimidated if some people have seen some of this material. Let me come back, and we're going to thank you for the great ideas. I'd now like to do a very simple simulation, continuing on the ideas that we had developed a few minutes ago, which is I'd like you to fire up CompuCell. Even though we were talking about Mechanica a lot, I would still like to fire up CompuCell. I'd like to ask you to fire up CompuCell, and based on the things that we just covered, I'd like you to build a simulation with 100 by 100 by 100 lattice, one cell type. You can call it anything you want, mesenchyme, cell one, anything. A surface area constraint with lambda volume of 2 and a target volume of 125. A surface area, surface area, a volume constraint. This is a typo, typical cut and paste typo. A volume constraint with lambda volume of 2 and target volume of 125. A surface area constraint with lambda surface of 2 and target surface of also 125. And then use Blob Initializer to draw a sphere of cells centered at 50, 50, 50 in the middle, radius 20 and initial width of 5. And if you use the wizard, that should take about 3 minutes to do. If you need, and so I'd like people to just see if the things we covered so far make sense. And then after that, we'll start doing things. So I'd like everybody to get started, and if you have CompuCell on desktop, that's fine. As I say, I seem to be having trouble screen sharing CompuCell Twit today. I don't know why it doesn't want to screen share properly when I use the desktop version. But you could run it in NanoHub as well. If you get stuck, I have a version of the simulation available as well. But why doesn't everybody start that? And as usual, I use the polling to let people let me know when you've got it running, okay? If there are any questions about what you need to do, let me know. And I guess I could do it, instead of using Wizard, I could do it by editing the code that we have here. Right. So that shows you, by the way, how much work it takes to go from 2 dimensions to 3 dimensions in CompuCell. And it also shows you a little bit, it's not a very good approximation of a sphere, but that's the Blob Initializer's approximation to a sphere. And if you have any problems getting this to work, feel free to ask. We've got Josh on the call and myself. We want to see the parameters that we have. So that's the basic simulation. And I'll let people think about that for a little bit, keep working on it. And let me tell you, well, let me wait. Let me see. There are 1 or 2 people. I want to make sure everybody's together on this. Okay, so far we've been playing with the XML for CompuCell. And now I want to turn to the other part of the simulation. And so to do that, double click on this. If you go to Twit, double click on this component that says Steppables here. Everybody see, if you have problems finding it, let me know. So everybody find the Steppables code here. Everybody's got it. Okay, good. So the Steppables basically is the place you can write Python code to manipulate all sorts of cellular behaviors. Essentially, the XML specifies the static properties of the simulation. That doesn't mean the things that the XML specifies are static. They're dynamic. They're cells, but they're their attributes don't change in time. The cells are the cell types they are. The initial configuration is what it is. In Python, we'll use Python to do things that we have to do events. At some time, you're going to introduce a chemotherapy. At some time, you're going to kill half the cells. At some time, you're going to do something else. And the basic structure that CompuCell uses is called a Steppable. And a Steppable is a class, if you're a Python person. It's basically a subroutine that is called every Monte Carlo step. Actually, you have some choice. You could change the frequency at which these are called. But let's walk through a Steppable and use it at least a little bit before the end of the class. So the first thing that you'll notice is that there's a frequency here. There's something called frequency equals 1. And that says that you call it once per Monte Carlo step. As I mentioned, you can't call it more often than that. It really should be called period rather than frequency. Because if you set the number to 10, you don't call it 10 times per Monte Carlo step. You call it once every 10 Monte Carlo steps. So it's an interval. I'll remind you that it's Python, so that indentation matters. And Twit will help you keep track of indentation. You will see that all of these Steppables typically have 4 sections. They'll have an init here at the top. And that's the place you put globals and things like that. It's called once at the very beginning of a simulation. You have a start function that is also called at the beginning of the simulation. And if you were going to write Python code to do initial configuration, to read in files, that's where you'd put it. And certain kinds of display and analysis go in the start function. Some of them go in the init, some go in the start function, and that's something we'll talk about. Then the step function is called every X Monte Carlo steps, as defined by frequency. And that is where you're going to do most of your work. If you want your cells to divide, to grow, to die, that's where most things will happen. And then, assuming that the simulation ends regularly rather than being interrupted, there's a finish function that you could use to write out data sets and do other kinds of things. Often that finish function isn't called because we'll hit stop in the player rather than letting the time clock for the CompuCell run out. Okay. So really, we're going to be focused on start and step and some things in the init. And all of the boilerplate here you don't really need to worry about. The one thing that you'll run into is CompuCell allows you to have multiple Steppables, and information is not automatically available between Steppables. And so if you have multiple Steppables and you want to get information between the classes, you have to do some tricks. For the moment, we'll do everything inside a single Steppable, and then you don't have a problem with that. The other thing you'll notice is that occasionally things will have selfs in front of them, and that depends on where they were defined, if they're local to the Steppable or not. Again, for the moment, just take it as a given that if you define your own variables, it won't have a self, and Twit will tell you when you need a self. That's a Python annoyance. Okay. Inside of the, we've discussed how to do things like define cell volumes and things in XML, in the Python, in the CC3D ML. We now are going to start learning how to do these things in Python. And so cells, in particular, also chemical fields and some other things, are Python objects in CC3D. And each cell is going to be referenced by a pointer, a name, a handle. And typically, cells will be identified by cell. But you could, in fact, give it any name you wanted. You could call it artichoke, as long as you're consistent. Each cell has its own unique identity, and you can have an arbitrary number of them. And cells have attributes. And those attributes, some of those attributes are for free. I said not much gets given to you for free. A few things get given to you, like cell.id. I told you that each cell corresponds to an index sitting on a lattice, and that index number is the cell's ID. It's an integer. And so cell.id will tell you what the unique identifier of the cell is. I mentioned that cells have a type. If you called the cell mesenchymal, it's mesenchymal. If you called it cell 1, it's type cell 1. We mentioned that cells automatically have a volume, how many voxels they occupy. If we have a volume constraint, we also have a target volume for each cell and a lambda volume for each cell. And the editor has all these. If you go to the editor and go CC3D Python and pull down that menu, you'll see this long list of predefined functions. And within that list, you have lots and lots of things that you might want to do. So if I want to know what the main cell attributes are, I go to cell attributes, and this will give me code for saying, how do I reference lambda surface? How do I reference lambda volume? How do I reference other things? And the main difficulty with Twinit, these menus, is simply remembering where things are, because there are a lot of menus. The good news is that every now and then there'll be something you need that's not in one of these menus. But in general, almost everything you need is there. So you don't have to remember the names and the functional forms. Okay. So one of the things that we're going to do a lot in CompuCell is do things to every cell. You'll look at every cell and ask, is it dividing or not? You're looking at every cell and ask, is it dying or not? You look at every cell and say, is it growing or not? And so CompuCell will keep an inventory of all of the active cells. That inventory won't include cells that you haven't created yet. It won't include cells that have died and disappeared, only the current existing cells. And that inventory is a list, and it has the not very exciting name, self.cell_list. And if you want to do something to all cells in that list, you use the classic Python iterator syntax, for cell in self.cell_list, : and that will create a loop that will iterate over every cell. Now, you might not want to iterate over every cell. You might want to only iterate over cells that are of a particular type. And you might want to do other things as well. And so if you go down to visit in the pull down menu, you could visit all cells. That's for cell and self.cell_list. Visit all cells of a given type will tell you how to pick ones that are only mesenchyme or only this or that. And the other ones are more elaborate things that we're not going to deal with, but you can iterate over the spatial volume for chemical fields. Cells can have substructure, and you can iterate over the substructure of cells and so on. But typically for cell and self.cell_list is the one we're going to use the most. And then we print here cell.id, and that'll print the identity of the cell. And if we go to the console, let's see, I don't know where the console is here. Not so easy to visualize it in Advective. It would print that information to the console. Okay. So the one thing we're gonna run out of time, but I want to tell you how to do one thing before we break, which is, so far we've been able to make movies, make pretty pictures on the screen. But if we're actually going to use it to do something scientific, we need to be able to do calculations. And so one of the things that CompuCell is quite good at is creating scientific plots. And I want to walk you through how to do plots. at how to add a plot to this simulation. And so again, we don't have to memorize these things, although some of them we look up in the manual because there are lots of options. But if we want to create a plot, we have to do three things. The first thing we have to do is create a window to display the plot in. Then we have to create a data series, which is going to be the data set the plot represents. And then we're going to have to create points and add them to the plot, to the data series. And so there are three steps. We're going to create the plot window. We're going to add a data series to the plot window. And those are going to be done in the start function. And then in the step function, we're going to add data points to that data series. And so if we want to create a dynamic plot, the first thing we're going to do is go to the start function somewhere. And you can do this, okay? And then you'll go to CC3D Python, go to scientific plots, and it says setup, and you'll notice it says in parentheses start function. It's telling you it belongs in the start function. If it's something strange where it goes in the step function or the init or somewhere else, it usually should tell you where it goes. And so I can hit start here. And it's going to paste in some template code. And you'll see that it has a couple of lines here. The first line is going to say self.plot window equals self.add new plot window. So on the right is a function that creates the plot window, the place you're going to put your plot. On the left is a handle that references that. And you're going to have to remember that. Plot win underscore win could be anything. It could be artichoke, but you have to remember whatever it is, you'll be using it repeatedly. And then within the plot window, you have to go to title, the name of the x-axis, the name of the y-axis, and then you have some options, which you don't need to specify, but you can, whether the x scale is linear, whether the y scale is linear, whether they're grid lines, and there are a bunch of those. Those you can look up. Some of those you'll get with a right click on the plot. Okay, so that creates a place to put a plot. You can have as many of those as you want. Each time you call one of these, it plots up a new window. You have to give each one of them a different handle. So if you have self.plot win underscore win, you want to do another one, you'd call it plot underscore win 2 or something else. Can't be plot underscore win twice. Then we're going to add two data series to our plot window. Self.plot win, handle we've got. Add plot, this adds a data series. The variable name, and how we're going to draw it. Lines, dots, there are a variety of options, what color it is, how big they are, and so on. And again, we can add as many data series to one window as we want. We have to give each data series a different name, so we couldn't add plot mvol twice. That'll cause a problem. Any questions about that? Okay, and it's boilerplated in here. In general, the defaults here are not going to be terribly useful, but they're not great. Okay. And then if we actually want to add something, information to the plot, we have to go into our step function and let us actually go into the step function and add to the step function some data points. So we go back to CC3D Python, scientific plots, add data point, and it'll paste in self._plot window, happens to be this. If we'd named it something else, we'd have to have a different name. Add data point to the data series M volume, corresponds to M volume here. MCS is time, cell.volume, cell.surface. Okay. Why don't we run that and see what happens? Can everybody follow along and try that? Let's see. See if it works. Use tile to arrange things. So I've got my image of my simulation here, and I've got my data set here. It doesn't look very pretty, and I can keep it running. And you can say immediately, see if you got something like that. Why am I getting such a mess? Well, the bottom one here is a mess because I've got lines turned on. The top one is not as much of a mess. So let me go back to my option here. I'm going to make this guy dots as well. And let me make them both a little bit bigger. You can play with this yourself. And again, if the screen doesn't redraw properly in NanoHub, pull on the corner of the outside box, and that should get it to redraw. Should be operative. Okay, there we go. Now, you'll notice I've got lots and lots of dots. The reason I've got a lot and lots of dots is that when I did my iterator, I'm plotting the volume of every cell and the surface area of every cell at every time step. That's a lot of points. So what I would ask you to do as the last little exercise we do in class would be, here I said add some code to plot the mean, volume, and surface as well as the individual ones. Plotting the individual ones isn't that useful, although you could do it. Why don't you try that? So what I want you to do now is use the idea of an iterator. You know the volume of each cell. So think of a way now to calculate the average volume of each cell and the average surface area of each cell. And then add a plot window that plots the volume and surface of every cell, but rather the average volume and surface of all the cells. Okay? And that'll be the last thing we'll do in class, I think, today. Once you can do something like that, you can essentially do almost any calculation you want. Clearly, the calculations may be more complicated, but to do an iterator over the cells, do a numerical calculation and display it is a big part of what you would want to do. I'd point out that if you right-click on the plot, it'll pull up a variety of options. Not 100% of these work. The package that's used to do the plots is a little buggy. But you could do rescaling of the plot dynamically. You can, transform doesn't work, I promise that. Most of those plot options don't work. Grid works. And you can also save the data set. So if you don't want to write it in code, but you decide at some point you want to save the plot, a right click will allow you to save the data in the plot. So is it clear what we're talking about here? And I'll do it along with you. I won't do it in a very elegant way, but why don't you people try to think about doing that little exercise? It's up to you whether you want to have it as a separate plot window or you want to replace the things that are currently calculated. I'll do it as called for, where it's a separate plot window. And I won't do it in a very Python way. If I were being Python-y, I'd use NumPy and do these all as vector operations. One thing you might want to know when you're doing a counter is if you're calculating an average for a value over the cell list, you might want to know how many elements it has. And it should, len of self.cell list should return the number of elements. So if that's something, you could also put in an explicit counter here. And again, I hope you're doing this yourself rather than watching me try to do it. I just want to make sure that I've got it. Oops. Well, so I hope you weren't following me if I've got a syntax there. Syntax error. Invalid syntax. All right. Let's see, what did I do wrong? Bullet point. Oh. Helps if the parentheses match. Huh? All right. Okay. How did that go for people?
I had to exit NanoHub and then re-enter, and I can't load the simulation. It's saved, but I can't load it.
Huh. What's it doing for you? Or not doing for you? So you see it, but when you try to open it in Player, it doesn't open. I mean, Twit it doesn't open.
It'll open in Twedit. It won't open in Player. I select the file.
So you go to, you're in Twit and you go up here and you say open in Player, and what happens?
Let me see. Session terminated.
Huh.
Or it's because the server disconnected.
That's disturbing. What about other people? Is anybody else, were people able to do that or not do it? Other people, how did it go for others? Are people still working on it? Collie, do you want to share your screen? I don't know what, I'm not sure what
Sure.
the issue is.
I started a new session. It looks like it's finally loaded. Oh, file.
Okay, why don't we just take a look at the code together and open it up in a second.
Oh, is this all empty now? Okay, I may have to.
Double click on lecture CC3D there.
Okay.
Okay, and now let's look at this code together. So, okay. So this doesn't have any plotting in it at the moment.
Right.
Okay, so let's look at the XML together. So click on the XML, okay. So you've got 100 cube, that's fine. Let's look at the cell type. Hold on. Okay, so you created cell 1, fine, okay. You've got a volume constraint, okay. And we've got a surface constraint, that's fine. Center of mass, that looks fine. Okay, so now let's go up here to this February 2 lecture .cc3d.
Should this here steppable be Blob Initializer?
Probably yes. Except that when you change it, it's not going to, the lines in it are not the same. So right click on, go above the blob initial, go to line 39 here. Right click, and now go to CC3DML up at the top. Second one, the pull down menu. I'm sorry if I forgot I'm not annotating.
Here?
Here.
Oh, okay.
Steppables. Blob Initializer. There we go. Okay, and so now under Blob Initializer, you could 50, 50, 50 is what you want. Radius of 20 is what you want. Width maybe of 5 rather than 20. 7. So you might change that. Okay, and then that's fine. Now we better get rid of that uniform initializer. So scroll down. And delete all of that. Not end CompuCell 3D. Yeah, good. Okay, now save all here. This one. And now right click on that and see if it'll open. There you go.
Okay. Yeah, thank you.
And if you want to make it 3D, you pull down here on XY. Okay. And you might resize here. Make that bigger. And then go to Window, Tile. There you go. Okay. Did anybody else have a problem? People able to get that to work? Okay. So I'll try to, since I never know quite how far we're going to get for the homework, I'll assign you these simple exercises on plotting that we were going to plan. And then that should give you some familiarity. Who was able to get the plots to work? Who tried it? Nobody's showing. Well, for kind I know you. Okay, Tyler, you got it to work. Fanbo, you got it to work. Great. Jeffrey, were you having a problem with it, or is it okay? Okay.
No, I got it.
Okay, you got it. Great. Okay, good. So tomorrow we'll write up a simple exercise based on plots, and then we'll try to keep thinking about coding and about projects. Okay. Anything you want to discuss before we break? I know it's a little late, but I'm impressed. Everybody's come up with some really interesting project ideas. Okay, well then, thank you very much, everyone. I appreciate your patience.